I want to make two observations to this author's audience and students. Some of them have already left, perhaps, even though there was such an interesting topic which Professor Gupta dealt with. One thing which you might have noticed, this is for the students, that Professor Gupta works on plant cytogenetics, mapping. He keeps up with what is the latest in the realm of biology. And that is the lesson which we should all learn, including the teachers. Nothing is fixed. We must continue to at least understand what is going on in the field of biology. My second observation is, I came here wrongly on the 7th of February. I thought it was 10th. So you can see that I can mess up things. So if I tell you certain things, don't take them for granted. But I had opportunity to speak to the first year and second year students, and I gave them a very detailed lecture on our work on the genomics of some of the classical species, particularly mustard. So I'm not going to speak on that today. I'm going to show you some slide on my other interest in the about agriculture, what is happening, and where is our research and what we need to do. So I'll quickly go through because the time is flying away, and we have to have a panel discussion also, and my very dear colleague A.K. Singh is to speak also on rice. So this is a shot of human migrations into Asia from Africa, and this happened from years have been given. The first movement was pretty long time back. There were many movements in New York, but it was much later. Why I'm showing this is that for much of human evolution occurred in Africa, and it's only later that the human population moved out. Next slide, please. Now, for all these years of human evolution, for most of the time, human beings were hunter-gatherers, foraging, hunting, living in small tribes. It is only after around 10,000 years back that plants were domesticated in various parts of the world. So our history starts, the empires, the civilizations, the modern world, all started after plants and animals were domesticated in certain areas. And why the domestication didn't occur earlier? Because in Africa there was no concept of larger society. There was enough of food and enough of animals, and it was only 10,000 years back that Earth started warming up. So the rivers started flowing with a lot of water, and the plant domestication and agriculture started in the river, along the rivers, including our Mohenjo-daro and Harappa, the Indus Valley civilization, because on the river side. These are the centers of origin of some of the cultivated plants. I mentioned Rabilau again because he, in my view, was one of the greatest scientists of our times. And some of the agriculture actually started in the Fertile Crescent. Unfortunately, this is what in this area, in the Euphrates River, Iraq, parts of Turkey, and because plenty of water was available, and barley, wheat, many other interesting crops were domesticated. Some of the crops were domesticated in South America, only a few in Central America, which included corn, which is a major crop, and in China also, and some crops in India. Next, please. Now the global population was about 1.6 billion in 1900, and today India is going towards that kind of population alone. If we look at undivided India, you will find that we have already crossed in this part of the world the population size that the whole world had at around 1900. So there has been tremendous explosion in population, courtesy of modern medicine, better hygiene and modern medicine. So how are we feeding so many people? That has been a miracle that has been brought about by agricultural sciences, by chemistry, by a lot of other mechanization, so on and so forth. Next slide, please. In spite of the fact that we had tremendous science and technology developments in the 20th century, it was really a century of great inventions and great scientific discoveries, including the whole field of molecular biology, which started in the middle of the last century. And you will see here that how many people died in famine, including the big Bengal famine. 7 million people died in that famine. The Chinese had even bigger famines, and look at where they have gone now after adopting science and technology in their country. In 1958, they had very bad famine conditions, and a lot of people perished in that. If you look at the Sustainable Development Goals, 2 of the major goals of UN are, which have been adopted by all the countries, no poverty, and most of the poverty exists in the rural side of India and the rural side of Africa. Urban poverty is also there, but a lot of poverty is because of the landlessness and no other alternative employment for people. And zero hunger is the other thing, which the world keeps on talking about, but is barely doing anything in terms of concrete action. Next one, please. So how did we meet in the last century? A lot of people predicted that millions will perish. Actually, they perished in China and in Bengal before our independence. But 4 things led to that we are able to feed today most of the humanity. Some overfeed and some reasonably feed and some underfeed, but at least we are able to not have big famines any longer. Fertilizers, they will wash process to fix the atmospheric nitrogen into ammonia, mechanization, plant breeding, and management, better management of the farm. So I will be concentrating a little bit on this to give you some little understanding of what happened. Some of you might have already read about it, but these days there is so much to read that one tends to forget some of the big lessons of even the last century. Next one, please. This is Norman Borlaug, who is considered to be the initiator of green revolution. Actually, the gene to make wheat dwarf was discovered by the Japanese and incorporated into their varieties. And when the Americans, US was occupying Japan after the Second World War, they found these dwarf wheats. They took these dwarf wheats to the United States, and at Pullman in the West Coast, where spring wheats are grown, American scientists incorporated these genes into their wheat and benefited from that. And Borlaug took it to Mexico because Mexico was facing huge hunger situation. Their wheat yields were very low, even corn yields were very low, and the US wanted to help them so that there is no mass migration from Mexico to the United States. And Dr. Swaminathan was one of the scientists who was very in the forefront in bringing these Mexican varieties to India and spearheading their adoption in India and also some of the initial breeding work which we went on. And I'm not really surprised that the government has finally decided to recognize him in Bharat because he did such a good job of introducing these materials into the country and nurturing Indian programs of translation. Next one, please. In rice also there is a dwarfing gene found. Now this was a lucky break. I call it the biggest jackpot that effect. It's good to find the gene with one good effect. But this gene dwarfs the plant, but it has also a pleiotropic effect that it led to better partitioning of the photosynthase from the leaf to the grain. And this is really part luck, but part breeding acumen to introduce it to the crop, the observation power. And even today, plant breeding requires very strong observation because you are selecting on the basis of phenotype. You are not depending upon huge data computer generated. Incidentally, I told the students on that day, and I must repeat it before I forget. This is my one advice to them. Don't be data shy. And if you have an opportunity to take a second subsidiary or second subject in your bachelor's degree, go for computer sciences. Because mostly what Prof showed you in the earlier lecture, you cannot sustain that amount of huge data without you being conversant with some programming, and you cannot add to the program also because you are computer language illiterate. So try to get some literacy in that, and that's my only suggestion to you. Others will emerge as you go along in your careers. Next one, please. This slide is partly vanished, but I wanted to show you that how much germplasm was used, how many processes were made to bring about this wonderful variety IR64. And this is not the variety that's grown today, because as we go along, there is huge amount of pest and pathogen pressure on the plants. Agriculture is not an easy area, because most of the world has turned urban, so people don't fully understand what are the big issues with agriculture. You have to take care of the weeds, you have to take care of the pests and pathogens. And you know, in human beings, so much research is being done on human health. Billions of dollars, much more than what is being spent on agriculture. And we are still prone to diseases and we run for medical help every other day. In plants, first of all, what we selected to grow in the early days or what we domesticated are all mostly self-pollinators. And a self-pollinator is a true breeder. Once you fix the variability, it's a true breeder. So if you go to Punjab, Haryana and UP during the maize or during the wheat growing season, you may have 2 or 3 varieties growing over 5 or 6, 7, 8 million hectares. And they are all very similar varieties and highly prone to pest attack and highly prone to diseases. And the most miraculous story as far as I am concerned is not the dwarf gene, but how wheat breeders have kept some of the diseases like rust and smuts and others away from the crop and has fulfilled the need of human nutrition by breeding. And that is the area I am sure Dr. Ashok Singh is going to talk to you about how much they have done for bringing these genes from some other sources into the varieties which are well adapted to India and which we relish to eat, like basmati rice. Next one, please. So, what was the miracle in the Green Revolution? You can see after 1960s how much the yield has increased in the cereal crops, particularly 3 crops: maize, rice and wheat. Rice and wheat due to dwarfing genes and maize due to hybrids. And you can see that the production has gone very high and the population, it has gone beyond the population. And that's how you are generating some surpluses now. While the land under cereals has been kept at about the same as it was in 1961, a little bit higher than that. So we are having more productive intensity, and that is where India is lacking. We have, except for wheat and rice, we have lower than world averages in all other crops, particularly legumes and oilseeds. We have done very badly in terms of wheat. Even though we are the maximum users of groundwater in the world, no country exploits so much of groundwater resources as we do. And fertilizers are subsidized and still you get low yields. I think this is a matter of great, should be a matter of great concern to everybody. Next one, please. So you saw some of the latest ones. I want to draw your attention a little bit to the past. Mendel's laws, Mendel's work rediscovered, as I told the students on that day. Mendel died unrecognized, nobody knew what he had done. Only when they rediscovered the Mendelian laws, they found out that this has been already done by Mendel. Then the chromosome theory, the chromosomes which Dr. Gupta talked about, all the chromatin is condensed and so on. And then the Watson-Crick model. The fastest developments anywhere in any science have been in biology. And the conversion of biology into products has been the fastest. Maybe in electronics, we can compare it to as much progress in rapid progress that has happened in electronics area. But this is cumulative knowledge. We started with genome analysis in 2000, and now the world wants to do the Earth biogenome. Incidentally, Dr. Gupta is very nice of him to summarize these things for students and for general readers. But India is not participating in the Earth biogenome project. Nothing has been done so far. So, what are the positives? What do we have in plant breeding today? We have we can do the old-fashioned phenotypic selection, which is still very relevant. We can use molecular markers to tag the important regions in the genome which are of economic interest. We can do genome sequencing has become a regular affair, as he told you. And in genetic engineering, which we are very reluctant to get into, and we have been fighting the case in court after the government decided to release whole transgenic mustard, which is about 20-year-old technology. And we are still wrestling with it, whether to release it or not. It has been released in Canada. Canada has about 8 million hectares under rapeseed with this technology, and nobody has found any deleterious effects whatsoever. And of course, in genome editing, there you can make null mutants in a very precise manner, which can be also very interesting. But Dr. Gupta told you the latest where you can put a gene at a particular loci in the plant through the genome editing enzyme. We have some negatives. Everything has to be bundled in the seed. Dr. Singh has one trait, I have another trait, somebody sitting in the United States has the third trait, and we don't like each other, we have commercial interests. So the farmer is not getting the best product, and that is the international challenge. They are saying international challenge is this or that. I say international challenge is to go over this intellectual property and to try to develop good seed with genetic resistance through genetics rather than spraying with agrochemicals. Generation time. There's not much you can do in this world, although there's a new concept of speed breeding due to LED lights that we have got now. You can change the spectrum and all that. There is linkage drag. You cannot get recombination at your desire, but that day may come when we can induce huge amount of recombination in the chromosome so that the genes can be transferred fast. And the chromatin area, which Dr. Gupta was talking about, discoveries there may lead to our finding some genes or some methods to have higher rates of recombination. And of course, number of loci. As we gain knowledge, we have tens of genes that we want to incorporate into our plants. And if in segregation you want 10 genes to be present in a plant, there is quite an astronomical number of plants that you have to screen. And of course, you cannot regenerate every variety, and some of the crop plants are very difficult to regenerate in vitro, so you cannot use genetic engineering and genetic editing on that. These are still some of the difficulties where people can work on. A bit on mustard. as I mentioned, every crop has pests and pathogens. These are the 5 huge problems with mustard. And this is stem rot, which is a necrotrophic fungus, which kills the stem tissues, and so no yield is. We use the whole yield in the plant. There is white rust and alternaria blight. There is not much time for me to tell you how much reduction in yield these diseases are. So you have choice. You can either spray pesticide or fungicides to control them, or you can incorporate genetic resistance from the land races or from the wild sources. And this is a root parasite, which can be only tackled. The only solution of tackling it today is glyphosate. So if you make your plant glyphosate resistant, this orobanchator cannot survive glyphosate spray. So one effective way of removing this root parasite, which is affecting about 1 to 2 million hectares in Rajasthan and other dryland areas. And then there is aphids, on which my good friend and student Arun Jagannathan is working on to find some clever ways of controlling the aphids. We are not, our group is not doing any work on aphids, we left it to him. Next one, please. Now, there is huge variability in mustard. We found two types, the Indian type here in green and the East European types. And we found that when we cross them, they are heterotic for yield. So there is potential of hybrid production to increase the yield in mustard, and that is what we wanted to exploit through this Barnes-Barska system, which is nothing but a pollination control mechanism. It doesn't increase the yield by itself. It is a method in which you can make sure that you have made sterile lines and they cross with the other line, the other parent can give you the hybrids which are fully fertile and the harvest can grow them. Next one, please. This is our work on mapping of white rust disease. We found 2 genes which we characterized, which we showed that they are the candidate genes by genetic transformation, and we gave it to all the companies without charging anything so that we can have white rust free mustard. But these 2 genes are not enough. Sooner or later white rust will find a way around it. So your gene discovery should go on. Unfortunately, there may not be more than 3-4 labs in the country which have done any gene discovery for disease resistance in any crop. Next one, please. So there is so much work to do. And this is all the traits that we have mapped on the chromosome through developing mapping populations. And these traits we want to incorporate into to improve the mustard lines further for better yields. Next one, please. Now, I don't want to talk to you about the Barnes-Barska system. This has been in the news a lot, and I don't have the time. But can you go to the next slide, please? Can you go to the next one? This document I will leave with one of your teachers, and the copy, you could have a soft copy of this on your computer and read it. I will urge you to go through it, because you should know what is happening, what are the frequently asked questions by the general population. You may also have those questions also. So this may be the best way of your knowing about the Barnes-Barska system and how do we intend to use it. Next one, please. As far as I am very much concerned about what we do in India. Of course, we should have our eyes open. I must tell you that whatever sequencing we have done has been done mostly in collaboration with Western labs. We started at South Campus with Professor Kilesh Tyag's leadership. The first project on a big crop, rice genome sequencing. It was a mega project in which our scientists participated and benefited. There is no harm in interacting with outsiders if your own interests are served. But today we seem to be very reticent about it. We don't want to get into collaboration or into meaningful collaboration where people learn something, come back to the country and take the science forward. China did it in a big way, earlier Japan did it in a big way. But we are reluctant. We think whomsoever we send abroad will only stay there and will never come back. But this is a false excuse. If you put value on your people, and if you tell people that there are opportunities for you back here, many people would love to come back to their country and serve it here. My student and now a colleague in National Institute of Scientific Research has recently developed the genome editing method. He knocked out the porters which the glucose, mustard has secondary metabolites called glucosinolates. These are not for our taste or anything. These are basically defense mechanisms, part of the defense mechanism against herbivory. Because the plant is sitting there and it is vulnerable to all kinds of birds, animals, large animals, small animals, insects getting at it. So glucosinolates are basically defense. And all over the world, people are growing low glucosinolate rapeseed, which they worked out through conventional breeding. We also did that. And this kind of conventional breeding leads to the plant having low glucosinolate both in the seed and in the leaves. So these plants are very vulnerable to herbivory because they have lost their defense mechanism. Naveen mutated about 10 of the porter genes, and which transfer glucosinolates from the pod wall to the seed. So now the leaves have full quota of glucosinolates, but the seed does not have it. And I think this is one of the most interesting developments in the field of plant biotechnology. And I hope that there will be more such developments in the coming years. Next one, please. So, what is our national goal in agriculture? We want low input, low water requirement, low input of other natural resources, and we want low monetary costs also in terms of fertilizer, irrigation, agrochemicals, reduction in agrochemicals, and so on and so forth. And we are spending so much on subsidizing fertilizer, subsidizing electricity. In Punjab, the government sometimes gives free electricity to the farmers. So they keep on flooding the fields, even unnecessarily. So we have to put value in the seed. And there is very little research money which is going into developing better seed, whether it is disease resistance or pest resistance. Because there are intellectual property issues, it takes, as I mentioned to you, plant breeding takes a long time. So sometimes people don't have the patience. People want results which should be available within the working span of a government rather than leaving it to posterity. So we require higher outputs and challenges before India in agriculture research, feeding an increasing population. Still, we have outstripped China. We are the most populated country in the world today. The number of breeding objectives in each crop needs to be met. Consolidation of global seed and chemical industry, which is a threat to us. There are 4 companies which may be controlling about 80% of the biotechnology and agrochemicals in the world. And unfortunately, we have very poor research in agrochemicals. We are good in chemistry, but we have never utilized it to discover maybe a single molecule which has been of value to Indian agriculture. Please correct me if I'm wrong. And patent tickets. The whole CRISPR-Cas technology, which is such a based on such a basic knowledge, and I don't think these 2 ladies who discovered finally the mechanism and got a Nobel Prize for that, they were so much worried about money because they did not file their patent in the fast-track manner. Somebody sitting in MIT did the work later, filed it in a fast-track manner and got the patent before then. And there was a big quarrel on this issue. And of course, finally, the myth of organic agriculture. You can do it in select areas, you can do it in limited parts of the world, but you cannot have it as a general practice. There is neither enough organic matter nor enough ability to collect it and convert it into a good manure to feed all the crops. Next one, please, and then probably the last one. I'm saying it and I say it to this audience also, particularly the young students, and please don't get disheartened. We spend too little on R&D in the country. This seems to be off our radar. I don't know why. As scientists, we have failed to convince successive governments in putting more money into R&D. And agricultural R&D is critical for food, nutrition, and environmental security. And we live in the times of ecological excesses. Although we know that in Soviet Union, I mentioned to you Babylon. I don't have the time to talk about Babylon, but I will tell the students, please look in the Wikipedia what happened to. He was the greatest collector of germplasm in the world. He gave the concept of origin of domestication, the domestication centers of the world. And what happened to him? Because Stalin didn't like his saying, he imprisoned him in Siberia where he died. What a great shame. And these kind of things can happen if the regimes are autocratic and fascist in their tendency. We have to guard against that. And scientific communities need to champion the cause of SNT. The students, you do your studies properly and well and look for a better future. Thank you.